Okay, we're not quite done with the abdominal cavity. This is the bladder. I dissected out to the scrotum. The testes formed up here in the body and migrated out and got a heck of a lot bigger. That's a testis. This is the structure that my fingernail's touching. This is part of the epididymis. There's a fat body there as well. So sperm and testosterone are made here. This is where sperm are stored. There's a tube you can't really see that goes in. Actually, there it is. Goes in and attaches underneath the bladder inside the prostate gland, which you definitely can't see. Now that's how the male system is set up. I didn't really want to sacrifice an entire rat to show the female system. So I'm going to put a pig down here. Oops. And show you the layout. And it's easier to see. The female system's pretty small anyway. It's much bigger in a fetal pig. Okay. This little thing that I'm pointing at and touching is the ovary. This little wiggly thing is part of the uterus. In pigs and rats, the uterus is Y-shaped. And so this is part of the uterus down here, kind of disappearing into the space. Here's another ovary. This tiny little tube here is the oviduct that carries developing embryos down to the uterus, where if you wind up with an implanted embryo there, you're in a lot of trouble. But what you've got in the lab is a way to compare the way the system is laid out in terms of location. Obviously, there's differences there. In terms of the appearance of the glands, there's differences there. And the associated tubes. I showed you the sperm duct and told you a little bit about the other stuff, and you've got the tubes associated here. So I left some boxes to fill in. Now, in our case, we're going to open up the testis. Where my little pointer went to. Not entirely sure what that is. That might be the epididymis. It's usually up at the top, but that's in the right place, but it doesn't look right. Like I said, doing live animals, or formerly live animals, you see a lot of different things. Okay, this is the internal structure of a testis. There's lots and lots and lots and lots of tubes. The sperm are made in the tubes, working the cells from the outside edge to a space in the middle where the eventual sperm are released. And then they're carried out and stored in the epididymis, and then they go up through the sperm duct, the vas deferens, to where they're eventually going to be ejaculated. So that's what the internal structure looks like. Now we're going up into the chest cavity, into the thoracic cavity. And we're going to get poking in here. These blunt probes are very useful for tearing at things. That kind of bumpy structure that I'm touching right now is the thymus. That is a particularly large thymus gland. Is that a whole class of white blood cells goes in there to mature, and they are then called T cells. We'll talk about those later. Over here, the lung is split up into lobes. Again, it's a space thing. And there's 1, 2, 3, 4 lobes over here. And 1, 2, 3 lobes over here. So your next question is, I just counted 4 on one side and 3 on the other side, and using the heart as a marker for what side of the body you're on, I've got 1, 2, 3, 4 here, and 1, 2, 3, which leads us to a bonus question of, you got an extra lobe on one side. What feature of internal anatomy, and keep in mind you're in the chest, there's not a heck of a lot of internal anatomy here, leads to the development of that. Keep in mind, this is an asymmetry question. The lungs are asymmetrical, so the question there is to think about what else in the chest is asymmetrical, and the thymus gland is not the idea here. Okay, your heart has 4 chambers in it. Is that, you can't, well, let's see. I'm gonna go a little bit out of order so I can show you. Normally I would be drawing this up on the board for the classes in the lab, but I should be able to actually show you in the heart itself. I can get the heart out of here quickly. Okay. Before I cut it open, it's very common for mammal hearts to have these flaps on the top. These are attached to smaller chambers called the atria. They're the receiving chambers where blood comes in through the veins. And under times of intense stress and the blood pressure goes up, these things will inflate like balloons to kind of hold the extra volume. Down here are the ventricles. These are the main pumping structures of the heart. The atria, when the ventricles empty out, they've squeezed. The atria just need to be powerful enough to put blood down here and reinflate them. So the atria are smaller. They hold the same amount of blood, but they're smaller and thinner because all they really need to do is push blood down into the ventricles. I'm going to cut this in half. I'm going to try to cut this in half. The chambers never are side by side by side by side the way they are in like a plastic model. This is the ventricle. This is the left ventricle. This is the right ventricle. This is blood, clotted blood inside the right ventricle. The right ventricle has more space over here. They kind of wrap around each other. This blood up here that's a blood clot. This is the left atrium. This little bit you can see is the right atrium. So you've got 4 chambers, upper atria, bottom ventricles. The right ventricle, the blood leaves here, goes out through arteries at the top. The blood goes out of the right side to the lungs. It comes back into the heart on the left side. The blood from the left side goes out to the body. And very quickly on the main artery that pumps out of the left side, there is a little side street that actually feeds the muscle of the heart. The heart does not get nutrients and oxygen from the blood it's pumping through. The lining is too dense to protect the muscle from the flow through. So it needs its own separate blood system. You can't really see that on a heart this small. There are blood vessels on the outside here, but they're not really visible on a heart this small. So we've got a couple of heart questions. Your heart is a big, powerful muscle working 24 hours a day. It needs a lot of oxygen to do that. It needs a lot of nutrients to do that. Nutrients come into the blood in one place and then travel from there to other places. The oxygen comes into the blood in one place and then travels from there to other places. If you read through the lab, it talks about how everything is connected. What I want you to tell me for question 2 here is, how is that system set up to make sure that the heart, if it's not first in line, it's really close to first in line to get nutrients and oxygen? And the answer to that is to tell me, okay, the nutrients come in here. Between there and where the heart gets it, it goes to other places. Where's it go? And keep in mind, it's not going to be a long list because the heart needs to get this stuff pretty quickly. Okay, let's see what we got here. Going into the respiratory system. That's a muscle. That. Okay, if the light is reflecting off of this well, there's ridges that you can see on this tube. This is the trachea, this is the windpipe. And this goes down and branches through bronchi to the various lung lobes. And those ridges are reinforcing rings of cartilage. And this leads into your next question. This has reinforcing rings the same way that a vacuum cleaner hose has reinforcing rings. And it's for exactly the same reason. And so that's your question. What are the reinforcements being reinforced against? And keep in mind, if you give me an answer that would not be true for a vacuum cleaner hose, you're probably giving me the wrong answer. This tube right alongside, right there, tilt it back. That is the esophagus. Usually, I guess I must have pulled it loose. It's usually attached to the trachea through small membrane, but that is what you're going to compare this ridged trachea to. So it's just an appearance comparison. Now, there's a couple of questions that involve the throat where I'm not going to actually dissect the throat. I wouldn't have had you dissect the throat, but I'm just going to point where there. The trachea would be coming out of a voice box. Up on the surface of the voice box would be the thyroid glands, and then when you get up into the throat on each side and up in the cheeks on each side, there would be salivary glands. I read an article this week that supposedly they've discovered new salivary glands someplace up near the nasal cavity. I'm going to be a little surprised if that holds up. But your saliva, the purpose of your saliva is probably not what you think it is. You take food into your mouth, and before you swallow it, you chew it up into little pieces. Well, if you've ever tried to swallow a little bitty pill, you realize your throat's really bad at swallowing little bitty pieces of anything. So in order to give your throat something to grab and swallow, your saliva is literally a glue to glue the pieces back together temporarily so that you can take this mass called a bolus, toss it into your throat grabs it, pushes it into your esophagus, and then your esophagus grabs it and pushes it down into your stomach. And that's the main purpose of saliva. Now, this is the beginning of your digestive system, so anything we can do digestive, we're going to try and get done as quickly as possible. We do actually produce an enzyme in our saliva that starts the process of breaking starches down. Starches are pretty big molecules, fairly simple, but eventually you need them broken down to sugars to absorb them, and there's a lot of energy there, so that's what you want to do. We have other things that are much harder to digest that if we could, we'd want to get an early start on, even though the final process of absorption would happen down in the intestines. Lipids, I talked about how they don't like to dissolve in water. We have to do funny things with the blobs just to get them into a position where we can digest them. But we don't go after lipids with enzymes from the saliva. Proteins, much bigger, much more complex molecules than starches that also are not that easy to digest. You literally need enzymes for every little part of attachment parts in a protein. We don't go after proteins with saliva either. That's your next question. Why do we leave the lipids and the proteins alone? And the answer isn't because we take care of them later, because that isn't really an answer. The answer is, why not start early? Why not start up in the salivary glands with saliva to start that process of digestion? Okay. I know I say okay a lot. We're going into the head. So the first thing we got to do is get the skin off. I used to work with a guy who had to get organs out of deer in Tennessee. When you take a deer, you take them to, you have to check them in a place. And there's often science people there who will open your deer up and take certain select samples. So he would spend entire weekends dissecting deer in these stations. He'd come back just soaked head to foot in blood. Okay, this is the skull. A little bit of membrane still there. See, when you use gloves, you don't have fingernails to clear the membranes off. You can see that the skull has seams in it. Cross seam there, straight seam. You can't really see there's another cross seam here. What we're gonna do is try to get the brain exposed by getting the skull off of it, but not chopping the brain up doing it. Nice robust pair of scissors is good for this. And the seam is your best entry point. Now we're gonna get around the edges. You're not gonna be able to see me do this. Oh, it actually comes off fairly easily here. Now, rat brain, unlike when we do fetal pig brains look remarkably like human brains. Rat brains are a little bit more linear. They tend to have, instead of the second part being mostly tucked underneath the back of the brain, they tend to be more in line with each other, where the cerebrum tends to be in the front and the cerebellum tends to be in the back. Also, a rat brain doesn't look quite as human as a pig brain does. Okay, this is the front processing part of the brain. This is the cerebrum. This is where memories are stored and decisions are made and responses are coordinated. Sort of. Responses are planned, let's put it that way. Back here, this is trickier because this is a much thicker part of the skull to get through. Oh, that's a nice one. This is the cerebellum. You're actually cutting off your own light. Very ridgy. Is it yours would be much rounder than this and it would look kind of like a stack of plates. But when the brain decides, the cerebrum decides, okay, we need to do something, your cerebellum goes, okay, boss, and it decides what muscle needs to do what in order for the response to happen. So if your brain decides, your cerebrum says, we need to run away, then all of the messages to your muscles that are involved in running away are put together and sent out from your cerebellum. And tucked under your cerebellum and connected to the spinal cord is the medulla. You can't really see it. If I went after it, I would just mangle it trying to get it out, so I'm not going to show it to you. And you don't need to describe the appearance in the lab. But the medulla is kind of handles all the stuff that's always on automatic, like making sure your digestive system is moving food and your heart's pumping and your lungs are going. Okay, those are all the parts of the lab. You do have a question about the brain at the very end. And there are some other questions that I think I went past that are functional questions. Let me look back through. I didn't notice them as I went because I've been trying to do the cutting. Yeah, you've got a couple of hard questions that are a little bit tricky. But there isn't really anything in the dissection that's going to help you answer that. So, but that is the dissection part of this. And so this is where you're going to get the answers of how things look. Is a lot of the other stuff has to do with figuring things out. When we typically work in groups, we don't always have everybody cutting. So I want a bunch of brains working on other questions to figure things out. And my command to my lab groups is always, I'm fine with you helping each other out. Somebody over there gives me an answer. What I want you to make sure you do is you put your answer down in your own words. Because you have to think it through to put it into your own words. You have to sort of understand it to put it into your own words. Where if somebody just puts their lab next to yours and you copy it, That just tells me you can transcribe English. So for those of you that are in contact with one another, remotely, if you want to help each other out, you can do that, but I don't want two answers that are word for word exactly the same. Or are word for word something you decided, I can't answer this, and I looked it up on the internet. Most of the time, it's not gonna answer the question I asked. I tend to ask unusual questions that the internet doesn't address. But even so, if you do find something useful on the internet, put it in your own words. It proves to me that you maybe kind of understand it, and that's the purpose of me doing that. Okay, that's it for the rat lab.